In our decomposition chamber, we put in half a banana peel, some twigs, 5 blades of grass, compost, and 3 mealworms. In our terrestrial chamber, we put in 3 sweet pea seeds, 2 blades of grass, 1 leaf, 3 radish seeds, 3 potatoes, 1 nightcrawler, and 2 crickets. We used compost and non-fertilized soil. After several weeks, we noticed that there was no plant growth from our sweet pea seeds, so we added in carrots and 7 mung bean seeds. Our soil was also poor, so we added fertilized soil. With this addition, after a couple of days, we noticed a significant change in our terrestrial chamber with an abundant amount of sprouts from the mung bean seeds. In our aquatic chamber, we put in 4 mosquito fish and 1 bundle of elodea. We also used water from the turtle tank and a couple handfuls of assorted blue gravel. Since 2 of our fish died early on in the experiment, we added 2 more mosquito fish to replace them. We also decided to leave the 2 dead fish bodies in the aquatic chamber so they could decompose and add more nutrients to the water.
In the beginning of our eco column, our aquatic chamber was healthy. The elodea was green and full of life. We also had 4 healthy mosquito fish. Over the next few weeks, the elodea slowly decayed and our fish died off. We realized our carrying capacity of fish in the aquatic chamber was 1. The water at the end of our eco column was green and murky. There were dead fish as well as dead and decaying plant material floating around the chamber. As the fish died off, our 1 survivor had plenty of nutrients and oxygen from the elodea, making a successful aquatic chamber. The pH of our water was always 7 throughout the entire experiment. Overall, our aquatic chamber was mildly healthy. We now know, with the amount of elodea we had, only 1 fish can live in order to make our aquatic chamber successful. In the terrestrial chamber, there was no evidence of any life or growth in the first few weeks of our eco column. In the beginning of our terrestrial chamber, we had crickets, seeds, grass, leaves, and potatoes. Over time, everything except for 1 leaf completely died and decayed. Our terrestrial chamber was a complete failure during this time, until we added nutritious soil and seeds. After adding good soil, the nitrates and phosphates in it allowed our seeds to flourish over the next few weeks. We had a thriving plant life, and by the end, we had a well-balanced terrestrial chamber. Our decomposition chamber was full of many items to decompose as well as very nutritious compost soil. In a matter of 3 weeks, all of our products decomposed thanks to good mealworms acting as decomposers as well as good nutrients in the soil from the compost. At the end of our decomposition chamber, we were left with a healthy beetle, which is a product of our mealworms, and overall we had a very successful decomposition chamber.
The turbidity of our water in the aquatic chamber stayed relatively the same until it got extremely murky after about 6 weeks into the experiment. The nitrates in our eco column dropped about 3.5 weeks into the experiment, but then gained stabilization after about another 3.5 weeks. The temperature in our water stayed between 22 degrees Celsius and 19 degrees Celsius. In the beginning of our experiment, our dissolved oxygen levels peaked at 10 ppm about 5.5 weeks into the experiment, and soon gained a stabilization of 6 ppm after about 6 to 7 weeks into the experiment. Our phosphates declined 5.5 weeks into the experiment, went up slowly for a few, and now appear to be decreasing. The pH of our water was always 7. Throughout the lab experiment, the only major things that we changed were the soil in the terrestrial chamber and adding 2 fish in the beginning after the first 2 fish we added died. We changed the soil and added new seeds to hopefully produce some kind of vegetation and increase oxygen levels in the chamber. We added more fish in the beginning, hoping our aquatic chamber could support more than 1 fish, which we found out it could not.
We noticed that as the turbidity levels in our aquatic chamber rose, our observation of murky water worsened and worsened. As the dissolved oxygen levels in our aquatic chamber rose, our fish began to die off. We also observed that as the dissolved oxygen levels were very low, that our fish were very sluggish and were finding it very hard to breathe. At first, when we had 4 fish, we had our lowest dissolved oxygen levels, and after the first 2 died off, our oxygen levels shot up to 10 ppm. At this, we noticed that our fish became more lively and engaged. This then led to a stabilization where we had one fish and a stable level of 6 ppm for our dissolved oxygen throughout the ecosystem. After the addition of more nutrient-rich soil mid-project, we noticed both a jump in our phosphate and nitrate levels, and a much healthier aquatic and terrestrial chamber. Once our nitrate and phosphate levels rose, we noticed our plants began to grow and our Elodea became less wilted.
3 of our mosquito fish died because there was only enough food and nutrients for one mosquito fish to survive. Our crickets died very quickly into the experiment because it did not have enough food to eat and it was unfamiliar with its new environment, which was considerably cold because of the classroom's air conditioning. Our worm died because it was put in poor soil that lacked nutrients. If given the chance to do this experiment again, we would use different soil with a higher nutrient content. We would also have put in one fish to not go over the carrying capacity of our aquatic chamber. We suggest next year's class to choose the best quality soil possible and to start off with few fish in the aquatic chamber and to add more if needed to test the carrying capacity of the chamber.
The 3 reasons why there are differences in the eco columns are: 1. People use different materials, such as they have different soils with varied levels of fertility, which controls all vegetation growth. The second is the ratio of fish to Elodea. With more Elodea and less fish, the oxygen level is higher, which controls how long the fish live. The third is the amount of water because the Elodea and fish depend on it.
One food chain in our aquatic chamber was the dead fish decompose, which created more nutrients for the Elodea to feed on. The alive fish then ate the Elodea. The other food chain was the algae grew on the nutrients from the dead fish nutrients and the artificial sunlight. One food chain in our decomposition chamber was the leaves, twigs, banana peel, banana, decomposed, and the mealworms ate the nutrients from the compost and the decomposed material. The other is a mealworm grew into a beetle and ate the leftover decomposed nutrients. In the terrestrial chamber, the plants took in the artificial sunlight, carbon dioxide, and nutrients from the soil, and through photosynthesis created oxygen. Also in the terrestrial chamber, when the crickets and the nightcrawler were alive, the crickets fed on the potato bits and the nightcrawler fed on the nutrients from the poor soil.
A biogeochemical cycle is a natural process that recycles nutrients in various chemical forms from the non-living environment to living organisms and then back to the non-living environment again. Oxygen. Carbon is absorbed by the plants in the terrestrial chamber, and through photosynthesis, oxygen is produced. This oxygen will sometimes diffuse into the aquatic chamber, and in the aquatic chamber, through photosynthesis, the Elodea absorbs the carbon respired by the fish and converts it into more oxygen. Carbon. Carbon is created in the aquatic chamber through aerobic respiration. Once the Elodea gives off its oxygen, the fish breathe it in and through aerobic respiration convert it back into carbon. Nitrogen. Nitrogen was added into our ecocolumn in the soil. The bacteria in the soil, through nitrification, breaks down the nitrogen and the plants use the nitrogen to grow. The nitrogen is then flushed into the aquatic chamber by the rain and is used by the Elodea to make amino acids that the fish survive on. Phosphorus. Phosphorus was first added into the terrestrial chamber through the soil. As phosphorus does on a larger scale, phosphorus in our ecocolumn also traveled from our terrestrial chamber, or the Earth's crust, to our aquatic chamber, or the ocean. The phosphorus moved from the aquatic to the terrestrial by runoff. The water we poured through our ecosystem carried the phosphorus from our soil and deposited it into our aquatic chamber. These phosphates helped our Elodea thrive and continued to provide nutrients to our water. Water. In our ecocolumn, water undergoes the entire water cycle. The water we add is precipitation, and as the water flows down the chambers as runoff, it moves the other chemicals with it. Once the water ends up in the aquatic chamber, it evaporates and starts the cycle over again.
Some differences between our eco-column and others in the class are that our terrestrial chamber seems to have the most abundant vegetation growth because we added in fertilized soil halfway through the experiment. Our aquatic chamber is also less murky compared to the others, and our decomposition chamber completely decomposed because of the compost and mealworms we put in. Some similarities between our eco-column and others in the class are that our water in the aquatic chamber became more murky compared to when we first added water, the amount of Elodea decreased over time due to decomposition, and that each eco-column had less mosquito fish than the amount that was put in the beginning of the experiment.